translation. So translation is the latter part or second part of the protein synthesis. Protein synthesis actually starts from transcription, that is formation of mRNA by the DNA. So one of the strand of DNA acts as template, and the functional part of DNA that we call as a gene. So in that part, on one of the strand of DNA template, mRNA complementary strand of RNA is produced, and that is mRNA. So this mRNA, it comes out of nuclear pore of the eukaryotic cell and comes out in the cytoplasm and search for the protein synthesizing site, that is the ribosome. In case of eukaryotic cells, ribosomes are attached to the endoplasmic reticulum, while the mRNA, in case of prokaryotic cell, as nucleus is not membrane bounded, mRNA once produced, it directly goes into the cytoplasm. So here in this lecture, we will try to understand the process of translation. So after formation of mRNA, how the proteins are produced after reading the specific message which is there on the mRNA given by the DNA template. So that process actually is called as a translation. So in translation, proteins are produced, and these produced proteins are as per the instruction given by the gene, or which is present on the mRNA in the form of blueprint. We can say on mRNA this information is printed, and this is read by the ribosomes, and accordingly in the form of codon, the specific codon specify specific amino acid, and these amino acids, they are gathered together, and in between two amino acids peptide bond is formed, and this produces polypeptide chain, and this polypeptide chain forms the protein, and this protein is responsible for formation of characters. So as per requirement of the organism, or as per the requirement of the cell, DNA provides mRNA, and mRNA produces via mRNA proteins are produced in the cell system. So here we can see the process of translation. What is the meaning of translation? Genetic information contained in DNA template strand is transferred to mRNA, and this formation of mRNA is called as a transcription. Now we will see the definition of the translation. So after transcription, translation process starts, and this translation process we are looking towards, and the details we are finding out. So translation can be defined as formation of polypeptide chain of amino acids during protein synthesis according to the direction contained in mRNA is the translation. So what are the requirements of translation? If translation is to occur, what things are necessary for this process? So during translation process, important one are different types of RNAs. So transfer RNA, messenger RNA, ribosomal RNA, and also ribosomes are essential. At the same time, different amino acids they are necessary. Various enzymes are also involved in it, and various protein factors, they are also required for this, for neat completion of the process of translation, which is the second part of the protein synthesis, we can say. So first, transcription, where mRNAs are produced, and followed by the translation, where the formation of proteins according to the instructions given by the gene, so that occur. Now we have enlisted just now the different requirements in the form of different RNAs. Three different RNAs we know: mRNA, tRNA and rRNA. Also ribosomes, also different types of amino acids, various enzymes and various protein factors are necessary for this purpose. So one by one we will see some details of these required things. So firstly, ribonucleic acid, that is RNA. So RNA almost resemble in structure closely with the DNA, but has some differences. So difference lies between RNA and DNA. We'll see in brief. So in case of RNA, there is ribose sugar, whereas deoxyribose sugar is there in the DNA. In case of RNA, uracil is the base, and in case of DNA, instead of uracil, thymine is there. RNA is single stranded and DNA is double stranded. These are some of the differences. Otherwise, the polynucleotide strand, which is single stranded, is made up of nucleotides. These nucleotides are made up of what? Base, sugar, here ribose sugar, and phosphate. So such nucleotides, they come together and forms a polynucleotide strand. So actually RNA is polymer of ribonucleotides, in which there are 4 nitrogenous bases like guanine, cytosine, adenine and uracil. Here hydrogen bonds are absent, but whenever there is formation of loop, or when the single stranded polynucleotide strand loops around, the formation of hydrogen bonds, temporary hydrogen bonds, takes place over there. Now we will see different types of RNAs and out of that, first transfer RNA. So what is the role of transfer RNA and what is the structure of transfer RNA? We'll go, or we'll look towards those things. So here you can see the hypothetical structure called as a cloverleaf model of tRNA or transfer RNA, abbreviated as a transfer RNA. So this thing, the hypothetical structure, you can see over here. So in this, tRNA is for transfer, T for transfer, and transfer of what? Amino acids. From where? From cytoplasm. To where? To protein synthesizing site, that is ribosome. And this work is done by this tRNA. So 20 different tRNAs are there for 20 different amino acids for carrying them from cytoplasm to the protein synthesizing site, that is the ribosome. So some details we will see related with tRNA, how transfer RNA is abbreviated, that I have told you as tRNA, like this it is abbreviated. It is also called as a soluble RNA. So take S of this soluble and call it as a soluble RNA instead of tRNA. So both the names, they are there for this type of RNA. So this RNA or tRNA, it constitutes 15 to 20%, 15 to 20% of the total RNA of the cell. It is smaller than that of mRNA and rRNA. See, out of three, tRNA is smallest one and it has about 73 to 94 nucleotides. In all, 73 to 94 nucleotides are there in this single strand. Just see, it is looping around, and that's why we can see the structure just somewhat like a double stranded, but actually it is not double stranded structure. Instead, it is single stranded. So if one pull this part and another part apart, then you can have the single stranded structure. So see here, this is the clover leaf model, and in this clover leaf model, the details we will see. Actually, see here 5 prime end, here 5 prime end, okay, and this 5 prime end, at the 5 prime end there is a base, either cytosine or guanine. Then go towards this thing here in this direction. Actually, this sequence, it is called as a acceptor stem or acceptor arm. Then see here, this is the D arm and it is folded around, and this is loop 1. Here you see loop 1, and this is D arm where hydrogen bonds are produced in between the bases. Then D arm followed by anticodon loop. So this is the anticodon loop, and see the anticodons over here. Anticodon loop. This is anticodon arm, and here a loop 3. See here loop 3. This is loop 2. Loop 3 is of variable arm. We are going to see the details later in the discussion. After loop 3, there is loop 4. Loop 4. See this loop 4, and then the acceptor arm, which ends here somewhere, and the specific sequence of bases at 3 prime end. This is the 3 prime end, and this is 5 prime end. So 3 prime at 3 prime end, the adenine is the base, then comes cytosine, another cytosine base, and later here adenine or guanine, and this will depend on 5 prime end what it contain. If it contains guanine, then guanine or cytosine, and accordingly the adenine or guanine will be here. So this is the outline information of clover leaf model of the DNA. We will go into the details. I have just told you that tRNA is having about 73 to 94 nucleotides in its structure. Its molecular weight ranges between 25,000 to 30,000 Daltons. Remember, molecular weight is always measured in Daltons. This RNA, tRNA, is synthesized in the nucleus on DNA template. tRNA molecule is single stranded and looped or folded about itself that we have seen just now, and it looks like clover leaf. It looks like clover leaf, and this clover leaf model of tRNA is folded upon itself, resulting in 5' end and 3' pre-ends of chain which come near to each other. This 5' end and this 3' end, they come near to each other because of the self-folding. This structure, clover leaf model, has 5 arms that we have seen. We will see some details of these all arms. So, acceptor stem, see here this arm, acceptor stem related with this, it is also called as a acceptor stem or acceptor arm. It contains 5' and 3' ends. See this 5' and this 3' end, both they are there of tRNA molecule. Arm contains round about 7 pairs. See these nucleotide pairs, they are about 7 here. 3' chain ends with CCA. These 3 CCA base sequence. Amino acid actually binds to 3' terminal CCA sequence. To this 3' terminal CCA sequence, amino acid binds. So I will show diagrammatically. Amino acid binds like this to this tRNA, and further tRNA, what it does, it is having anticodon and it has sense which codon is specifying him or asking him to come over there and deposit the particular amino acid on the protein synthesizing site, that is the ribosome. So like this, the work of acceptor arm or acceptor stem, that is the amino acid is binded on this arm at the CCA sequence, and thus it is also called as a amino acid binding site. Acceptor arm is also called as a amino acid binding site. 5' end terminates with either G or C. That also we have seen. Then the next, here you can see the D arm. It is also called as a dihydrouridine arm. Dihydrouridine arm. So it consists of only 3 or 4 base pairs and possesses a loop, and that is the loop 1, and 8 to 11 nucleotides. Here only 8 to 11 nucleotides are there in loop 1. Next to that, anticodon arm. So this is the anticodon arm, which consists of 5 base pairs, and it is also called as a anticodon loop or loop 2, which is made up of 7 nucleotides. The middle of the nucleotides of the anticodon loop is known as anticodon. See, this is the middle portion of anticodon loop, and it is called as a anticodon. anticodon is complementary to the triplet codon in mRNA. Then the next term, which that is the variable arm, that is variable arm, also called as a loop 3. It shows variability in number of nucleotides, and number of nucleotides ranges from 4 to 21. Here 4 to 21. This is and that's why it is called as a variable arm. It contains 3 to 4 nucleotides or 3 to 5 nucleotides in general. Next arm, which consists of the 5 base pairs, and it is called as a loop 4, which is having 7 nucleotides, So this is what related to the structure of tRNA, that is transfer RNA, and we have tried to understand it with the help of clover leaf model. Okay, what is the role of this transfer RNA in protein synthesis? That we will see. I have told you that there are 20 different amino acids in the cytoplasm. They are necessary for the protein synthesis, and they should be carried to the protein synthesizing site, that is the ribosome. And for that, 20 different tRNAs, 20 different types of tRNAs, they are present in the cytoplasm, and they do the process of carrying of 20 different amino acids. So one amino acid has one particular type of tRNA to carry it, having particular anticodons. So in addition, tRNA has some other functions. So to carry amino acids from cytoplasm to the ribosome or protein synthesis site is the first and foremost work or role of this tRNA. Another, it can recognize specific amino acid present in the cytoplasm. That is also important thing. It can recognize specific amino acid in the cytoplasm, and to recognize codon on mRNA. That is also the work of what? This tRNA. At the time of protein synthesis, its anticodon should recognize particular codon, and then only in a specific sequence these amino acids will be incorporated in the polypeptide chain. So see how nicely the work of this tRNA remains in progress during protein synthesis. So just imagine a cell. Inside the cell there is cytoplasm. In cytoplasm amino acids are there, and to carry these amino acids, these vehicles, 20 different tRNA types, they are there, and they bind with the particular amino acid, and they start their travel towards the protein synthesizing site, that is the ribosome. Okay, then we will go towards the another type of RNA, that is ribosomal RNA, rRNA or ribosomal RNA. So what is the structure of ribosome? We will look towards the structure of ribosome at first. See the diagrammatic representation where 70S type of ribosome is here, 80S type of ribosome is here. So this is the smaller subunit, and this is larger subunit. Okay, this is association and these two are dissociated things. So larger subunit and smaller subunit, they have isolated, and some other details they are related with the ribosomal subunits that we will see later. Here ATS type of ribosome, 70S type of ribosome is present in the prokaryotes, while ATS type of ribosome is present in the eukaryotic cells. So this is the association of two subunits. This is dissociation of two subunits. Smaller subunit and larger subunit, they have dissociated over here. And then we will see some details related with the structure of ribosome. So they are ultra microscopic. What it mean? They cannot be seen with naked eyes. Ultra microscopic, only under electron microscope we can observe them. They are dense, they are granular particles, they are spherical in shape, and they have diameter of about 150 to 200 angstroms, and their length remains or ranges between 200 to 250 angstrom, and their breadth is about 150 angstrom. This is the measurement. Each ribosome is porous, hydrated, and composed of two subunits. Just now we have seen larger and smaller, larger subunit and smaller subunit. Larger subunit is dome shaped. See the shape over here. This shape is called as a dome shape. Larger is dome shaped. Here also larger is dome shaped. This is larger subunit of ATS, and this is larger subunit of 70S. Smaller subunit is cap-like structure. We can see here the cap-like structure of smaller subunit. Now how ribosomes are composed of what? Ribosomes are composed of what, that we will see. So prokaryotic ribosomes, they are composed of 60% of rRNA. Prokaryotic means 70S type of ribosome. It is composed of 60% of ribosomal RNA and 40% of proteins. 60 plus 40. 60% of rRNA and 40% of proteins. So this is the composition of 70S type of RNA. While in eukaryotic cells, I have told you ATS type of ribosome is there, and it is composed of 45% of ribosomal RNA, and remaining 55% is protein. So this is the difference between composition of 70S and ATS type of ribosome. Actually, S stands for, this S, it stands for Swedberg unit. This is a unit, Swedberg unit, S for Swedberg. Okay. According to the size and sediment coefficient values, derived from ultracentrifugation, these ribosomes, they are of these two types, that is 70S and ATS. Some details again related with the 70S type of ribosome now. So see here, this is 70S type of ribosome. They are found in prokaryotic cell that we have seen. In case of mitochondria also they are there. In chloroplast they are observed. sedimentation coefficient of larger subunit of this uh, ribosome is 50s, and this 50s larger subunit has molecular weight 1.7 into 10 raised to 6 daltons, while smaller subunit, this one, see this one, is 30s. Okay, here it is written 30s, and this smaller subunit is having molecular weight 0.95 into 10 raised to 6 daltons, and total molecular weight of 70s ribosome is 2.75 into 10 raised to 6 daltons. Okay, this is related with 70s, which is found in prokaryotes. Now related with the 80s, which is found in eukaryotic cells of plants and animals. One can find 80s type of ribosomes instead of 70s. So here sedimentation coefficient of larger subunit, see here it is 60s, and smaller subunit, it is having 40s. So total molecular weight of 80s ribosome is 4.5 into 10 raised to 6 daltons. These two subunits of ribosome actually remain held together with each other under low concentration of magnesium ions. But if magnesium ion concentration either becomes very less or very more, we can say, or high, the two subunits dissociate. So this is dissociated thing, this is associated thing, association of these two subunits. And remember here, this is not a mere sum of 50 plus 30 equals actually 80, but this is not the thing, so don't get confused. 70s ribosome has 50s and 30s subunits, while 80s ribosome has 60s and 40s subunits. So when dissociation and association of these ribosomal subunits takes place, that I have told you. Now we will see ribosome, which is a protein synthesizing site, and these ribosomes, they contain two different binding sites. So see here, you can see this is the first, see here, see this is the first binding site and this is the second one. First, see here, P site, peptidyl binding site, and A site. So these two sites, here it is a smaller subunit, this is a larger subunit. So related with the P site, it is also called as a peptidyl tRNA binding site. Peptidyl tRNA binding site. This site holds tRNA molecule. Actually, peptidyl binding site holds tRNA molecule which is linked to the growing end of polypeptide chain. So this is the growing end of polypeptide chain. Now related with the A site, amino acyl tRNA binding site. This is peptidyl tRNA binding site. Now amino acyl tRNA binding site, it holds incoming tRNA which is charged with amino acid, which is carrying amino acid. So it will come here at this site first. Actually, these ribosomal subunits, they are synthesized in the nucleolus and then they enter in the cytoplasm. These subunits attach to the mRNA to form polyribosomes. See, this is the mRNA. This strand is mRNA, and like this, smaller subunit and larger subunit, they attach over there, and there is formation of the polysome or polyribosome. Both the terms are used. The structure is polysome or polyribosome. In the cytoplasm it is formed. Now we will see related with the role of ribosomes in the protein synthesis. So actual role, it is the site of protein synthesis, that is their role. So protein synthesis takes place here only. 5' end of mRNA is first attached to the smaller subunit. See, this is the smaller subunit, and suppose if it is a mRNA, so 5' end, it attaches to the smaller subunit for the first time, and larger subunit contains enzyme called as a peptidyl transferase or peptide synthetase, which is involved in the formation of peptide bond between two adjacent amino acids. Ribosomes also contain about 50% of ribosomal proteins, which are known as protein factors, and protein factors are also necessary at the time of protein synthesis. The interaction between tRNA, amino acid complex, and interaction between tRNA and mRNA is coordinated by ribosomes during protein synthesis. mRNA strand which passes between the two subunits, there is cleft in between, and through this cleft or tunnel, the mRNA, it passes. Somewhere here we can show it like this. So this passing mRNA through the two subunits of ribosome, it should be protected, and this protection of mRNA from the action of enzymes of nucleus called nucleases is necessary. there are some protein digesting enzymes even like proteases, and larger subunit do the protection of these proteins from the protease enzymes. Now we will see mechanism of translation. Actually, up till what we have seen is related with the types of RNAs and their role. Now we will see the mechanism of translation. We know DNA determines as which protein is necessary for the cell. The information or this message is contained in the DNA for protein synthesis. DNA transfer this information of protein synthesis to the mRNA during the process called as a transcription. The information in mRNA is in the code language in the form of triplet codons. The language of triplet codons in mRNA is expressed in the language of specific sequence of amino acids to form polypeptide chain of protein, and this process is called as a translation. Thus, the process in which genetic information contained in mRNA directs the sequence of specific amino acids during protein synthesis is called as a translation. So, translation process is a complex process and it includes various steps. It begins with activation of amino acid, then transfer of activated amino acid to tRNA molecule, initiation of polypeptide chain, then elongation of polypeptide chain, and lastly termination of polypeptide chain. So different steps we can once again enlist. Activation of amino acids, and due to activation they are transferred to the transfer of activated amino acid to the tRNA, and then initiation of polypeptide chain formation, that is protein chain formation, initiation, then its elongation, we call it as a chain elongation, and lastly somewhere this chain should terminate, and the event or step is described as a termination, chain termination or termination of polypeptide chain. Now we will see the first step, that is activation of amino acids. You know 20 different amino acids they are there in the cell which are necessary for the protein synthesis. Amino acids occur in the cytoplasm. They are in the inactive state and they are incapable of attaching to the respective tRNA. So 20 different tRNAs they are there to carry the amino acids, and these amino acids, if they are in inactivated form, they are not attached to the tRNA, and therefore each amino acid should be activated, and this activation is as a result of or due to the ATP. So this ATP, see here, amino acid plus ATP plus enzyme, amino acid synthetase enzyme, and as a result of that amino acid adenylate complex is produced and phosphates are released. So see once again, for the first time amino acid gets activated, and activation is by the ATP, that is energy is provided from the ATP, that is adenosine triphosphate, and this process requires activating enzyme amino acid synthetase. 20 different amino acids they are there in the cytoplasm, and also 20 different amino acid synthetases. These enzymes are also 20 different. Then transfer of activated amino acid to tRNA. Transfer of activated amino acid to tRNA. Here activation we have seen. Then the transfer. So during this transfer, see here, amino acid adenylate complex plus tRNA, and it forms tRNA amino acid complex. This is tRNA amino acid complex. Then adenosine monophosphate and enzyme, they are released. So here, while giving description, we can say that activated amino acid, that is amino acid AMP, binds to the acceptor arm of its specific tRNA. Then it reacts with tRNA in presence of enzyme amino acid tRNA synthetase to form amino acid tRNA complex, and AMP and enzyme amino acid synthetase are released. So this can be seen in this reaction. Now third step, that is initiation of polypeptide chain. Initiation of polypeptide chain. So see the requirements of initiation. For initiation, what is essential? So you can see here, this is smaller subunit, and to this smaller subunit, this is the mRNA. So at its 5' end of the mRNA, it attaches to the smaller subunit of the ribosome. So here smaller subunit and mRNA. Then in presence of GTP and initiation factor 1 and 2, they are required for this attachment, and like this complex is formed. So see here, the protein formation initiation takes place by selection and transfer of first amino acid to ribosome. It requires a small subunit of ribosome, then amino acid tRNA complex. Just now we have seen how it is formed. That amino acid tRNA complex is necessary, smaller subunit of ribosome is necessary, Then mRNA is necessary, and initiation factor we have seen, that is also essential, and energy source in the form of GTP is also necessary. So related to the initiation factor 1, here initiation factor 1, it is present in prokaryotes and called initiation factor 1, initiation factor 2, also initiation factor 3, and in case of eukaryotes as M1, M2, M3. Now formation of amino acid methionine, the first amino acid in the polypeptide chain of protein is methionine, abbreviated as a MET, M-E-T, MET. So see here that complex FMET is here. This is tRNA. It is carrying this amino acid. The complex is called as a FMET tRNA. It settles on the P site of the ribosome. Now formation of the amino acid methionine. So I have told you, first amino acid in the polypeptide chain of protein incorporated always is methionine in case of eukaryotes, and N-formyl group, that is CHO group, is added to the amino group, that is NH2 group of methionine to form N-formyl methionine in case of prokaryotes. So always in prokaryotes N-formyl methionine, and in case of eukaryotes methionine will be the first amino acid during or in the protein at the time of initiation, protein formation initiation step. This amino acid is incorporated, and this process of addition of either this methionine or N-formyl methionine is called as a formylation. Now formation of mRNA 30S initiation complex. In prokaryotes, ribosomal subunit attaches to the 5' end of the mRNA to form mRNA 30S complex. And this process requires initiation factor 3. See here, initiation factor 3, and magnesium ions are also necessary. 30S ribosomal subunit always attaches to the first codon of mRNA. First codon on mRNA is always AUG. That also you remember. First codon on mRNA is always AUG, and that is why it is called as a initiation codon. or start codon, so which is necessary for initiation step. This AUG codes for formylmethionine, N-formylmethionine in case of prokaryotes and methionine in case of eukaryotes. Now activated YEP-MET attaches to the tRNA to form YEP-MET tRNA complex. In this complex, tRNA consists of anticodon UAE, which is complementary to AUG. That is the initiation codon of mRNA. YEP-MET tRNA complex now binds to mRNA. See here, it binds to mRNA to form a complex called 30S pre-initiation complex, and the process requires initiation factor 1, initiation factor 2 and GTP. YEP-MET tRNA complex attaches BITES anticodon, which is the anticodon UAG, to the initiation codon, which is that AUG, at 5' end of the mRNA. Now attachment of the larger subunit. We are here up till now attachment of the larger subunit to the smaller subunit. So in case of prokaryotes, larger subunit is of 50S. So this 50S ribosable subunit attaches to the 30S pre-initiation complex. So this is all pre-initiation complex. So to this pre-initiation complex, this larger subunit attaches. This complex includes, see here, 70S ribosome, larger and smaller. 70S, then there is mRNA, then tRNA, and in addition YEP-MET. See this one. So during this process, means when these two subunits they unite, so there is release of initiation factor 1, initiation factor 2 and initiation factor 3, they gets released. In prokaryotes, first YEP-MET tRNA comes to lie and attach in the peptidyl site or P site of the ribosome. Now elongation of polypeptide chain. Elongation means addition of amino acids one by one to the first amino acid, that is either methionine or N-formylmethionine. So how this elongation of polypeptide chain takes place, that we'll see here in this figure By referring this figure, one can understand the process of elongation of polypeptide chain. So, for elongation, first event of requirement, that is the elongation factors. So, elongation factors, they are required. For example, EFTU, EFG, they are required in case of prokaryotes, while elongation factor 1 and elongation factor 2, they are required in case of eukaryotes. After requirement of elongation factor, binding of amino acid tRNA to the A site of ribosome. Amino acid tRNA binding site we have seen in case while studying the structure of ribosome. So binding of amino acid tRNA to A site of ribosome. So tRNA of second amino acid, tRNA complex, means AA2 tRNA 2 recognizes the second codon on mRNA, second codon of mRNA, and fits into the site of the 50th subunit of ribosome. Here, anticodon of second tRNA is complementary to the second triplet codon of mRNA. So, see here how this thing takes place. See, like this, in this direction, mRNA will move. This is the direction of mRNA, while direction of ribosome is opposite direction. So, like this, mRNA will further move in this direction. So, next triplet or next codon will take A site. Accordingly, tRNA will come over there and will be having the specific amino acid, and like this the process continues, and addition of amino acids takes place, and at the last, termination of polypeptide chain. So termination means end of polypeptide chain formation. Means incorporation of new amino acids in the growing polypeptide chain will be stopped. So this thing happen when ribosome reaches the triplet codon, which is a stop codon or end codon, which will be present at the 3' end of the mRNA. So these termination codons are present on mRNA. They are either UAA or UAG or UGA. So these three are the termination codons. These are also called as nonsense codons, as they do not code for any amino acid. So whenever ribosome come to these codons, the further elongation of polypeptide chain stops, and the process is called as a termination. So termination codons actually gives signal to the ribosome to attach with terminating protein factor called as a release factor or F. So in case of prokaryotes, these release factors are named as RF1, RF2 and RF3. GTP also helps in termination of polypeptide chain. So this is how we have seen the process of translation in brief.